Blood-feeding insects, particularly mosquitoes, ticks, and kissing bugs, spread numerous diseases. This method of disease transmission is through a combination of what is known as pathogen-host vector interactions. Insects become infected with the pathogen, a microorganism that causes disease, after biting an infected host, and then spreads that pathogen to an uninfected host or animal by biting them. Dr. Donald Champagne, a research professor at the University of Georgia, is studying ways in which blood-feeding insects assist the pathogen's entry into a new host by blocking natural immune defenses.
All vertebrates have defenses against blood loss. So we've got platelet aggregation and clotting, and the other thing is that the activated platelets release factors that cause the blood vessel to constrict at the injury site. So that's vasoconstriction. Anybody who feeds on blood has to deal with these three problems, okay? and that is accomplished through factors in the saliva, proteins or peptides that are injected as they're feeding or as they're probing and looking for the blood vessel that degrade these wounding signals, prevent platelets from recognizing the fact that an injury has occurred there, and so the platelets don't aggregate together, they don't form a plug. Usually, also in the saliva are molecules that actually cause blood vessels to dilate, so vasodilators. So, everything is sort of turned on its head. Instead of the normal wounding responses, the blood vessels open up and you get more blood coming in, and so they can feed more rapidly. Okay?
Without these natural responses to blood loss, the insect saliva actually assists the pathogen's entry into the body.
You've inhibited the wounding responses, clotting and vasoconstriction and all of that, and that you've also inhibited the immune response or modulated the immune response in the skin where the saliva is going in. Now imagine a parasite. If let's say your mosquito is infected or your tick is infected, right? When it goes into the host, it's not going into a normal piece of skin with intact immunity and intact hemostasis. It's going into a very compromised environment. So the vector is not doing all of this to help the parasite. It's doing it because it has to be able to feed. Okay? But the parasite, the pathogen, can hitchhike on that, right, and take advantage of it to get established.
So it has been shown that these salivary proteins counteracts the body's hemostatic reaction, or the reaction of blood loss, as well as suppress and kill lymphocytes, the cells responsible for killing pathogens and bacteria via our immune system. This knowledge has led to some of Dr. Champagne's most recent findings about mosquito saliva.
This lymphocyte killing activity, I've identified candidate protein that I believe is responsible for that activity. And right now, I'm working on trying to develop an expression system so that I can make recombinant protein. I can do certain kinds of manipulations of that protein to try and, first of all, to confirm that it is the right protein. Have I got the right thing that's responsible for this particular activity in the saliva? And then, what part of the protein is important, right?
With all of this in mind, Dr. Champagne's findings are vital to limiting disease transmission by these blood-feeding insects.
I'm interested in vaccines in the longer run. I'm working on some proteins that inhibit certain immune activation of certain aspects of the immune system. And so the idea is that when I've really narrowed down or nailed down that I've got the right proteins, then those would become important candidates for vaccination. The idea is if we can understand blood feeding, what is happening during that process, it might suggest or might lend information about what is happening during the transmission process.